Hello, everybody. Happy Monday. This is the last day of the third marking period. Well, technically, tomorrow it switches over. I'll be going through the grade book today. A couple of you had emailed me and messaged me about grades, uh, and we'll go through. I know some of you start a little bit slower, then you do better at the end of the quarter, and those grades that you get towards the end would bring up the ones earlier. So I would say your grades should be your least of your concerns now for the third marking period. I will be looking at them carefully and uh, helping you out as much as I can in areas where I think people, maybe aren't where they should be. If you had one lower grade earlier, and it's definitely different than the type of grades you usually have, I'm sure I'll be able to find some um, Corona extra points to throw around. So uh, I'll update the grades hopefully all today and be checking your grades today and tomorrow. The grade window doesn't close to the 15th, even though we're going to switch to the fourth quarter. So there'll be some time for us to email back and forth and talk about your grades. But, okay, so moving on, we're going to run through Meiosis really quickly here. I have a assignment in your Google Classroom, and that is to make a meiosis slide presentation like you did for mitosis. If you can, I would love for you to include the video like you did for mitosis, because I haven't seen you. One, it would be better for you to do, and two, I haven't seen all of you in weeks now, and I would love to be able to look through your slide presentations, see you, hear you, and have you do it that way. It would be better for you to learn it, talk about it, go through that whole process. Because the more you have your hands and minds on the topic, you'll be able to understand it. So if you could shoot a video at home and include it into your slide, that would be great. Now, when we did the mitosis, I asked you to put it in your website and just show me your website. Now, because I don't have access to you showing me your website, I do want you to add your slide presentation to the assignment. So when you do open up the assignment, you're going to click on that little paperclip to add it from your drive, add the Meiosis slide presentation, and then I can look at them through the Google Classroom. Also put it on your website, make a Meiosis page, put it on your website, put other Meiosis stuff on there. Again, just because the more you mentally manipulating this material, the more you're going to remember it. Because you will see it again in ninth grade in bio or in your pathways in biology. If you take just pathways classes, you'll see meiosis again. So even though you're technically not getting a grade on this yet, and if we switch to the fourth quarter to grades, we will be getting a grade on meiosis. You want to learn how this works in general, okay? So let's go through meiosis. One of your slides you want to have is something that looks like this. I'm not sure where it's at here. These four terms make up a slide. So you have these, because these are new definitions: meiosis, homologous pair, gametes, and crossing over. In meiosis, we're getting four genetically different reproductive cells. These aren't body cells, they're reproductive cells. A homologous pair is a pair of chromosomes that are genetically similar. The gametes, two genetically different reproductive cells, or four of them, or I guess I should say genetically. Just should say genetically. You end up with two at the end of meiosis one. And crossing over, that's when the chromosomes literally cross over or lay over each other during meiosis. Let's go through this. Meiosis, we know it starts with interphase, and that's the same as mitosis, where the DNA is duplicating, the organelles are duplicating, the cytoplasm is increasing. It's basically growing and developing. Nothing is different there. Prophase 1, and we're going to go through it twice. Prophase 1, all the DNA is duplicated. You go from 46 to 92. All the organelles are duplicated. The nuclear membrane starts to break down. Spindle fibers and centrioles form. The only real difference here is when the chromosomes move out into the cytoplasm, they start to find their homologous pair. And that's what we've modeled here. Chromosomes that are about the same size, shape to another one, and they'll pair up with them. Okay, metaphase. Everything is pretty much the same as mitosis, except when they line up, it's not a single file line. Everybody's lined up with their dance partner. Everybody's lined up with their homologous pair right here. Okay, metaphase is over. Spindle fibers are attaching, and metaphase is over as soon as they start to pull apart, which we know is anaphase 1. Cytokinesis also begins. That is where the cellular membrane begins to pinch in, and we get this cleavage furrow. Now, in this case, the thing that's different than mitosis is the homologous pairs are separated. So I have a full X-shaped chromosome going that way. I have 46 of them going that way, and 46 of them going that way. 46 chromatid. 23 X's going this way, 23 X's going that way. Each has 2 chromatids, so that's 46 going this way and 46 going that way. Telophase 1, cytokinesis continues. The nuclear membrane returns, the nucleolus returns, and in each nucleus I have 46. Cleavage furrow deepens. All right, cytokinesis is complete. I end up with 2 genetically different gametes. Because of that crossing over and swapping of genetic material back in prophase 1, we're going to have 2 genetically different cells here, 46 chromatid in each. Then the process starts over again. We do not do interphase again because we don't need any more organelles. We don't need any more cytoplasm. We have enough. We just need to split this DNA some more. So you go right into prophase 2. Nuclear membrane breaks down. Chromosomes come out. Centrioles and spindle fibers reform. Now, metaphase 2 looks a lot like metaphase from mitosis because we have this single line. And you can imagine now this half is going to be pulled this way, this half is going to pull that way, which means I'm going to have 23 chromosomes going this way, 23 chromosomes going that way. And we'll see that in anaphase. Cytokinesis 2 begins, and there is those 23 chromosomes going towards each pole. Telophase 2, cytokinesis 2, cleavage furrow deepens. The nuclear membrane returns again. Nucleolus returns again. And you see now all this single V-shaped chromatid in each nucleus. Cytokinesis 2, we end up with four cells from the original 46 to 92 chromatid, now split up 23 in each. Okay. And that's that. That's the Richter scale. That's how to measure earthquakes. So you can design however you want your slides. There's going to be quite a few slides. Unless you do 2 on each, then there'd be a little bit less. Have yourself a drawing of some kind, a modeling of it, some just words of description. Very basic. You should be describing most of it. Remember, most of the description should come with you. It should be a very simple drawing, and then you provide the description of what's going on. Very basic. Like mine, very basic and poorly drawn. Let's see. There was something else I wanted to mention today, and not much has changed. I often forget things. Meiosis, slide, attach it, video if you can. Nah, I don't remember. I have one more batch of podcast science facts I'm going to do. And if I remember what the other thing was, I already talked about grades. I don't remember what else it was. It's going to be a beautiful day today, 60s and sunny. Get outside if you can, and enjoy that sunshine. All right, everybody. That is it. I'm stalling because I can't remember what the other thing I wanted to tell you was. If I remember, I'll tell you in the podcast. Okay, everybody. Have a good one.